question is going to be for Dr. Lee. How do you evaluate discriminatory dissolution methods?
Hi, this is me. To answer this question, I think it really evaluation of discriminability of the method is a case-by-case basis. First, you need to understand the drug product and the release mechanism, and to determine whether there's any formulation or process attributes that expected to impact the drug release. For example, sometimes for poor solubility drug substance, maybe particle size, or for ER formulation could be the content of polymer in a matrix formulation. And then based on your understanding your product, you can determine what could be a meaningful range for that attributes, either formulation or process. And in that case, I think we do expect that the dissolution, if the risk is following medium level, that is we target the method can discriminate meaningful change in these critical formulation or process attributes. In that sense, for example, you may target to see a difference between like if you use a micronized particle size, if you go larger to like 40 micrometer, this type of thing, you may see a difference in the dissolution. Or if you already like, for example, if the formulation is osmotic pump system and you have the membrane coating with the polymer, the coating weight again might be a parameter that you may consider for the discriminability. For example, within some range, you may define a range like within that range is acceptable, and but if you try to say out of the range, manufacture batch out of range, perhaps your method should be able to discriminate on that part. I hope I answered this question.
Great, thank you so much, Dr. Lee. And we have another question for you. How would you proceed with dissolution development if the drug substance class is unknown?
Yeah, that is basically, if you start to develop a dissolution method, you need to investigate some basic biopharmaceutical properties of drug substance. For example, the solubility, permeability. Sometimes you can, for generic drug product, you can refer back to the IOD, the label, or regarding the ADME, and to see at least you can estimate whether the permeability is high or low. If it's really unknown for permeability part, that is also, I think you may go with the worst scenario, like a low permeability, try to see how to proceed. But solubility, if you do not have the solubility data, you definitely need to evaluate, provide the data in your summation to determine whether it's highly soluble or poorly soluble drug substance.
Great, thank you so much. And we have another question for you. How would you classify risk for BCS Class 2 and Class 4 drugs?
Yeah, this is I think that is for IR solid oral dosage form, and in that case, BCS is one consideration. You know, BC class 1, BC class 2, BC class 2 and 4, they are poorly soluble drug products. But some of the BC class 2, you still maybe like some acid drug product. You may also have high solubility in in a physiological relevant pH range of the small intestine. In that case, you may also have the dissolution testing, that if the dissolution showing rapid in under pH 4.5 to 6.5, you may be able to classify as low risk. But if you do not meet that, you may go to see whether you have a CBA. That is all for all the low permeability poorly soluble drug. We all go that route. First, look at if you have a CBA available. If not, if even you have a CBA available, can you detect that, clearly detect it, or and controlled? If you can do that, then the risk is still falling in medium risk. Otherwise, we'll go to high risk or very high risk. So BC class is only one consideration. You still will need to consider dissolution as well, and overall I think the therapeutic risk for this drug product, for narrow therapeutic risk index drug product, you may need additional consideration on the risk assessment.
Great, thank you for answering these questions. And we may come back around to you, but right now we're going to go to Dr. Pai. So Dr. Pai, for the ANDA registration batches, if process validation activity is performed on the same batches, would you review reports and confirm if any additional studies need to be performed prior to commercial launch?
Thank you for that question, Lisa. So I do understand the question is seeking to clarify whether when a process validation report is submitted, that would get reviewed. So from our perspective, the information that you submit within P3.5, we would review that as supportive to the application and for the exhibit batches with regards to the content therein and any concerns it might bring up. But with regards to it supporting your process validation activities, that would be a decision you would have to take. Based on our review of feedback, you may have to revisit the information that you provided with regards to maybe process parameters or additional controls or sampling, and reconfirm that the intended validation activities reflect the commercial process. And so therefore, the agency does not comment or provide feedback on the actual validation activity as part of their review. It may be looked at during inspection. Thank you.
Great, thank you so much, Dr. Pai, for answering that. And we have another question for you. Can you comment on manufacturing considerations that may need to be included in a submission in order to support site transfer, or for including additional drug product manufacturers?
Sure. So one of the things to keep in mind is that your process development activities identified a range of unit operations and supporting parameters. Now, if you are considering that they be executed at site A, you've done additional scale-up studies supporting the equipment at site A. If you're thinking about moving this or implementing this in addition at a site B, you may want to take a closer look at a variety of factors, starting with your equipment, your controls, any other additional considerations that may be unique or different between the sites that have a potential to impact product quality, and factor that into your site transfer activities to support addition or inclusion of a second site. So really, you're taking a comprehensive view of the inherent characteristics of the product and the process, and then ensuring that each site is capable of executing the intended process to deliver the quality attributes that are expected in the finished drug product. Thank you, Lisa.
Great answer. Thank you, Dr. Pai. And we have one more question for you right now. Is the role of OPMA confined only to reviewing process-related information on an ANDA application? And does it actually also extend to FDA 483 responses? If the 483 responses are reviewed, what does OPMA look for in the 483 responses?
Sure, great question. So OPMA reviewers do assess the process, facility, and as my colleague Marla mentioned, the microbiology supporting a drug product application. With regards to the facility assessment or the integrated manufacturing assessment, there may be recommendations for inspections, including pre-approval inspections, or during the review of an application, there might be surveillance inspections that happen that inform the agency about the capabilities of the manufacturing sites in the application. So as part of our ongoing review through the course of the application review, we do look at the 483s that might come up, the responses to the 483s, particularly if there was a pre-approval inspection conducted, and understand how those citations from a quality perspective, from a compliance perspective, have potential to impact product quality. There may be instances where concerns identified in a 483 are being intended to be resolved or remediated through either additional studies or through additional controls. And in that case, our review overlaps, and we may seek to have appropriate revisions or additions or controls be reflected within the application as well. So there is definitely an integrated assessment that happens should there be a pre-approval inspection, and appropriately consideration is given to any other inspections that occur and the related deficiencies or 483s that might have been issued during the review period. Thank you.
Great. Thank you so much, Dr. Pai. And with that, we're going to move on with the questions for Dr. Stevens-Riley. So the first question is, what size breach does the agency like to see for positive controls, such as a needle or a capillary breaching the stopper?
So currently, the size breach is there's no specific size given. This would depend on what is your container closure system and what type tests you're using. You might receive different certain parts of the agency may want to see studies with breaches that are at the 20 micron, 10 micron level. It depends on your study, and there needs to be justification for the type of breach positive control that you were using in your study. And there is a very good educational material in the USP 1207 chapter about creating positive controls and how to determine the size breaches that best work with your test and your scenarios.
Great. Thanks for the answer and the reference. So we have another question for you. If equipment such as autoclaves or etc. are identical in make and model for multiple fill lines, is it necessary to submit the 3 most recent validation reports for all equipment on each line?
So this is a little bit complicated because it depends on if you have 2 pieces of the same equipment, but they are going to, let's say, sterilize different materials, then what you are validating is the ability of that equipment to sterilize the particular load that's in it. So you need 3 successful runs with a BI to validate that that piece of equipment is sterilizing that load. However, if you have two identical pieces of equipment, the exact same autoclaves, that will sterilize the exact same load, then it is possible to validate the load in one autoclave, but do less studies, but you need to demonstrate equivalency in the ability of those autoclaves to at least heat or do some heating. So you may not have to do full validation, but you've got to demonstrate the equivalency of those two pieces of equipment and their ability to reproducibly and consistently heat and sterilize.
Thank you so much. We have another question for you, Dr. Stevens-Riley. What is the frequency of a media fill study required for a production line for each product?
So we expect media for initial qualification of a filling line, 3 successful runs are required, and thereafter it is expected to you requalify every 6 months, usually on the line. And there is a great aseptic processing guidance that the agency has that describes more of the expectations on media fills and qualifying lines for aseptic processing.
Great, thank you so much. And we have another question now. We're going to cycle back to Dr. Lee. Recently, for a couple of products, FDA requested to develop discriminatory dissolution method, where the FDA recommended the dissolution method or USP listed dissolution method, and it was followed for an ANDA submission. In what instance does FDA prefer a generic company to develop a new dissolution method rather than using the FDA recommended dissolution method or a USP one?
This is a very good question. Actually, FDA dissolution database or USP method is only considered as starting point for dissolution method development for generic drug product. It's not considered as FDA recommend method for this drug product. As you may know that the dissolution method is often formulation dependent. So sometimes you develop a method apply for especially for ER formulation. You develop a method for osmotic pump system. It may not be really good for matrix-based formulation. So in that sense, I just suggest you, if you have FDA database method or USB method available, you might first try with that method to see how this method perform. You may look at the discriminability, and if you think the method really discriminates, you might start using this method in your submission as a QC method. But we expect to see the discriminability of the method for medium risk level. If you could not see that discriminability, you might develop a new method. Also, the development also need to consider the decision to whether it's low risk or very low risk. Very low risk, we go to FDA guidance method. It's usually just 500 ml 0.1 anhydrochloride as a medium, and then very low risk that you might have limit dissolution method development information to support your method. But if you go to high risk or very high risk, you may need some kind of modeling work or IVIVC or IVIVR to support your method.
Great, thank you so much for that answer. And we actually have another question for you, Dr. Lee. For the solid oral drug products that are classified as very high risk, where there is no linkage established between the QC dissolution and the in vivo, how does the agency determine the appropriate acceptance criteria, specifically for commercial batches with respect to time points?
Yeah, this is a good question. For very high risk, we do not expect many IR product may fall this area, but for ER formulation maybe. So in that case, you might have in vivo dissolution to support your method, to develop IVIVR or IVIVC to support the biopredictive dissolution method. However, some cases you may not have that IVIVR or IVIVC. If you already tried and you can provide the data, still you cannot have that relationship. That is one reason you might consider whether the method really good. You may need to develop another method. Another part that if your method still can show discriminability upon some major like CBA or something formulation, a critical formulation or process parameter that you can see your method can discriminate on that part. And based on the discriminability, based on your in vivo studies, and we can set up the clinical not really clinical in that case. It's still maybe it can accept criteria based on all these data to assure within that range the drug can have the BE to the IOD and to your target drug product.
Great, thank you so much, and appreciate again our audience submitting these great questions, and glad we can take time to answer them. So our next question will be for Dr. Pai. Is DOE needed for the products manufactured for a few well-known steps?
Thanks, Lisa. So design of experiments is a very handy and useful tool. But however, the agency does not want to be prescriptive. There are many other alternatives to establishing product design and process selection and process design based on understanding of the risks related to the process. So we definitely encourage you to provide your rationale for the selected maybe process parameters or controls for your process. I just wanted to share that. Additionally, with regards to sampling, there is flexibility as well with regards to how you select sampling sizes or locations. What we really are looking for is that you have understood the risk through well-designed experiments. I explored the range of operating parameters you might see or encounter during commercial operation, and established your process and sampling plan accordingly. Thank you, Lisa.
And thank you, Dr. Pai. We have another question for you, and it is, can 3 registration batches can that data be considered as process validation if there are no changes in the intended batch size or process?
I really would refer you to the process validation guidance. Within our guidances, we're not prescriptive with regards to the number of batches. Depending on the complexity of the process, as well as the familiarity your site has with the unit operations, you may need more batches. However, it is up to your facility to establish your validation protocol and be confident that it is reflective of the commercial process. Thank you.
Great, thank you so much. And our next question will be for Dr. Stevens-Riley. And that question is going to be, for a terminally sterilized product, is the QBR for terminally sterilized products required as part of the ANDA submission?
No, I provided the reference. The QBR is a guideline that came out many years ago of the type of information to include. It just, if you look at it, you will see that it's a very helpful way for you to understand the type of information to submit in your application. and it's arranged according to the common technical document. So you will see that document is at the time was the way that the microbiologist could convey to industry, this is the stuff we want to see, this is the stuff we review. So QBR, those references are simply to help you organize and understand what would be needed for an application.
Great, thank you so much, Dr. Stevens-Riley. We have another question for you. What is the FDA expectation on microbial hold time information for the unfiltered bulk and filtered bulk?
So there's not a specific hold time. Anything that you do should be justified with a study to demonstrate that there's not excessive microbial growth. And I want to say, could you repeat? I think there was another part of that in that question.
Sure.
Oh,
so
I see it.
Go ahead.
I'm sorry,
Yeah.
I can see it now. So it was on hold time. So the hold time before filtration, that needs to be justified. And if it's extensive, then there needs to be a study. And then once it's considered sterile, again, after it's been filtered, if it's being held, that time period should be justified as well. And that typically it may be included as part of the media fill studies.
Okay, great. Thank you. And we actually have another FDA subject matter expert joining us for our panel, and I'm going to introduce her quickly, and then we'll go ahead and ask a couple questions. So we have Dr. Pineiro-Sanchez, and they possess 20 years of experience in CMC assessment of generic products in CDER. She currently serves as the Senior Pharmaceutical Quality Assessor in the Division of Immediate and Modified Release Products 3 in the Office of Lifecycle Drug Products. She provides expert technical guidance in the pre-market quality assessment of generic products. Welcome, Mayra. And with that, our first question for you is, if DMF grade material is supplied as sterile, does the manufacturer need to demonstrate sterility before use?
Hi, thank you for that question. If there is a sterile drug substance and is supplied as such throughout the product and process development of the application, all the critical material attributes should be considered. For a drug substance, the applicant would have to confirm and test for release the drug substance, and that would include the testing of the critical material attributes such as sterility. In addition, these has to be also established and taken into context in terms of what the impact is into the finished drug product. So, so in general, at release, the drug substance, if it's sterile, that would be considered a critical material attribute, but that would be also can be established on application-based basis, but the general, in general, they should be tested.
Okay, great. And we have one last question for this panel, and it's going to go to Dr. Pineiro-Sanchez. If we have a worst case load of glass vials that we use for depyrogenation validation studies for all of our vials, can we simply state in the report that the maximum load of viral X is the worst case load of all vials?
I think that question for Marla.
Yeah, I'll take that question. I'm sorry. Yes, if you have the load of vials that your validation study contains, simply state the reasoning why that it is being used to represent your commercial production load. I think just stating its worst case a little bit more would be helpful. most of the time it's maybe based on weight or some other kind of information. That would be helpful, rather than just saying worst case. Tell us why it's worst case for the other vials, because sometimes it may not be that intuitive. So sometimes there's no study involved, but a rationale as to why it's worst case. Thank you.
Thank you so much, Dr. Stevens-Riley, and to all of our panelists for this great discussion. We're really happy that you were able to make the time today. And because we used our